Hello everybody, and welcome to this new appointment of the series of seminars SEM. It's a great pleasure to have with us today Nicola Toschi. Nicola is an associate professor in medical physics at the University of Vergata, a research staff and associate investigator at the A. Martinez Center for Biomedical Imaging of the Harvard Medical School. He has an interdisciplinary background in physics, mathematics, and neuroscience. His research is focused on the development of mechanistic, statistical, and intelligent models for understanding physiological phenomena, advanced image generation and processing, modeling of neuronal activity, nanoparticle physics, and bioactivity, and the development of software-hardware interfaces for real-time biomedical data acquisition. He has also a strong focus on MRI physics and ultra-high-field human MRI imaging, and regularly serves as a senior biostatistician, machine learning, and parallel computing consultant in fundamental biological biomarker research, genetic, epigenetic, and imaging, as well as an image acquisition and processing. He is the author or co-author of approximately 250 peer-reviewed publications in these fields. And now we can start with Nicola.
Thank you very much, Italia. Thank you very much, everyone, and thank you so much for the invitation. I'm very happy to be here. The slides are up, and thank you for that. So yes, in this seminar series, I'd like to, in this seminar in particular, I'd like to give you an overview of our work in the field of developing and applying also deep learning or machine learning methodologies to problems which are specific to neuroscience. As Italia was saying when she introduced me, I work at the interface between physics and mathematics and medicine. Both in my both appointments, and so this seminar will be partly translational. I hope this will be of interest to everyone. So I will just briefly outline the motivation for this work, and then give you some examples of the stuff that we're doing in this domain. I'd like to start with a quick example of the typical problem that you need to solve when you're confronted with biomedical, real-world biomedical problems. I'm sure you're all familiar with Alzheimer's disease, which has no cure. Now, the curious and the hugely impactful, both in society and on patients itself, problem is that as you age, you will lose some of your cognitive abilities, but there is a point where you will either just continue towards normal aging, or deviate towards Alzheimer's dementia, and nobody knows why this is, and there is no way to identify which patients would follow this trajectory or this trajectory in advance. Obviously, this has led to this is just an example, but this has led to the reckoning that there's no such thing as Alzheimer's disease. Obviously, patients who go this way, I don't know if you can see my pointer, probably not, but patients who take the light blue trajectory are different biologically and physiologically from patients who take the dark blue and then red trajectory, right? We just don't know how to extract that information from the wealth of data that we collect from patients. And this has led to the holy grail of medical research in the right now, which is called, as you probably might have heard, personalized and precision medicine, or some people like to call it P4 medicine, which on top of precision and predictive and personalized is also predictive and participatory. And there is a huge interest in the applied and also methodological research community in these topics, and hundreds of millions of dollars are being poured into this direction. The NIH has published roadmaps for how to integrate AI in medical imaging. The EU, European Commission, is constantly publishing funding calls which go in this direction, and so on and so forth. And the whole idea behind this is to realize, okay, these guys are all different. We don't know why, but the answer must be in the data and in the that we can already collect, because we have a lot of technology to collect data. And now it's all about how to extract this data and this information. For example, this slide is a little deformatted, so let me just skip to this one. We're currently able to extract multiple imaging modalities through simultaneous MRI and nuclear imaging, so structural and metabolic imaging. We can use radiomics, which I'm sure you're familiar with, to perform data augmentations, and that generates even more data. And normally, when we characterize a human being for these type of studies, we also have data from a bunch of other domains. Could be biomarkers from cerebral spinal fluid. It could be genetics, epigenetics, metabolomics. A big trend right now is blood biomarkers, for example, alpha-synuclein proteins in the blood, which is what you normally find in the brain of neurodegenerative patients, and so on. And that generates additional data, right? So the answer to why every patient, or the belief in personal medicine and personalized medicine, to the question, how is every patient different from every other one? Why does our one-size-fits-all approach not work? How can you make clinical trials more powerful? How can we separate these patient trajectories? Is very, very hot right now. And some people like to phrase it in terms of, how do you turn big data? Because by now we have public repositories, we have multi-center studies across the world which collect this type of data. How to turn this big data to smart data? And obviously AI is at the forefront of this sort of like worldwide research question, right? And there's wide room for application of both supervised and unsupervised paradigms. These are just very, very simple example. Of course, supervised paradigms would be extremely useful for decision support, so for characterizing single patients. Unsupervised approaches could be even more interesting, because currently there are a lot of clinical syndromes, so people that are sick with groups of disease of symptoms like dementias, like neurodegeneration, like autism, and things like that, which we know are formed of subgroups and substrata, which probably will separate, like I was saying before, in time. Some people will do well, some people will respond to therapy, some people won't, but we just don't know how to extract these categories, right, from the data that we have available. So this was just as a quick introduction and motivation to why this research is, in my opinion, interesting and important. So talking about the data that's available for these types of studies, apart from the type of data that I was mentioning before, which is genomics and biohumoral and even neuropsychological, basically sort of tabular data. Imaging data sort of sets itself a little bit apart, because when you start to characterize the brain, you can do this in a structural manner. So your data will be ordered on a three-dimensional grid. So you can use convolutional operators quite easily. Of course, you will have a parameter problem, because these images are at least 10 to the power 6, I guess, voxels they're called, so pixels, three-dimensional pixels in size. And so when you find yourself, a typical data set size would be from 1,000 to 40,000 patients. And so you will have both computational and, mostly computational issues. Or another very typical thing to do is to study what's called brain connectivity, meaning you subparcel the brain in different areas of interest, either a priori or through a data-driven approach, which some people like to call a tensor ICA. So it's a variation of ICA across both space, subjects, and time. And then you go and look how the crosstalk between these brain areas is characterized, and basically you end up with a weighted graph, right? And so the topology of the data is much less structured and much more individual, but it is very informative and also provides sort of an a priori data reduction strategy. And so these, which can be very helpful computationally speaking. Obviously, this is an oversimplification. There's a lot of other subtypes of data and of data structures that you can extract from neuroimaging and neuromonitoring data in general, whether it's MEG, EEG, whether the connectivity is dynamical, whether you deal with 4D images instead of 3D, and so on and so forth. But I just wanted to give you an overview of the main sort of starting points when you start designing learning architectures for brain data. So let me show you a couple of things that we have done in this field, in these fields. I'm just gonna give you a couple of examples and hopefully elicit some interest. So the first thing that I want to talk about is the problem that I mentioned in the introduction. How can you extract, can you take baseline data and extract, baseline meaning time zero today, and extract information about whether a patient will become an Alzheimer's case within 3 years or not, right? In the assumption that this information is in the data. Now, this is quite challenging computationally, and also it's also quite challenging because there is the assumption that the information is in the brain data because it is the brain that is suffering from the disease. But at the moment, there is absolutely no reliable way of doing this. And if it were possible, like I was saying, it would relieve a huge burden on a bunch of societal systems and subsystems, right? So the way we did this is to start from imaging data. In particular, we took a sample of about 800 MCI, Alzheimer's disease, and healthy control patients, which were publicly available. And we worked with this requires a kind of a little bit of an explanation, with the Jacobian determinant of the transformation, which warps this brain into a common space. So for every subject, you have this Jacobian determinant, which will tell you how much, as you know, how much the volume needs to be changed in a local manner, voxel by voxel, in order to turn single subjects into a standard subject. And in other words, it is a very sensitive way, in our opinion, to quantify cortical atrophy, how much the brain has shrunk locally. And we also used genetic data, and by genetic data, I mean just the expression of 2 or 3 genes, and neuropsychological data. And the task is, can we say, at time zero, this person is going to become an Alzheimer's patient and not, right? And so the way we approached this was with dual learning paradigm, where, the architecture is somewhere here, and there's a publication with this, but the idea here is to say, okay, preclinical patients and severe dementia sufferers are 2 extremes of a continuum, right? And within this continuum lie the 2 categories that we want to split into somebody that's going to convert and somebody that's not going to convert. So we train our architecture to learn both problems at the same time with a tunable parameter, which emphasizes one or the other problem more in the learning, right? This obviously will reduce overfitting, and obviously will also increase the number of samples that the network is trained on, if we can reasonably represent and believe in the hypothesis that this is a continuum, which is, I guess, a current consensus in the literature. People talk about the Alzheimer's disease continuum and ways to stratify it. The other innovation we introduced here was to use to I guess, recode 3-dimensional convolutional layers into separable convolutions in order to limit the number of parameters in these architectures. So as you probably know, separable convolution is a combination of a depthwise step and a pointwise step, and these have been shown to be basically almost with negligible difference as accurate as full 3D convolutions with a parameter reduction of about 20 times. And the code for this is available. And this has actually allowed us to process these images in the first place, which is a big problem, a big practical problem when you deal with neuroscience data. Yeah, so these are just details about the architecture of this particular study. And what you can see here, I guess I just want to highlight what you can see here is that we're using several, they're called modalities at the same time. So there's also an information fusion element to this. So the actual brain image, as well as the Jacobi determinant, as well as the clinical features, are going into this architecture with conventional stack blocks that are stacked, like in the different activation functions, dropouts, and so on and so forth. And there's different concatenation steps and two dense layers at the end, which basically solve the two problems at the same time: Alzheimer's versus healthy, and the harder one, a mild cognitive impairment who will convert and will not convert. And we're happy to report that this architecture has performed better than any other attempt in the literature to to predict this, even when we occlude part of the brain, which we like to interpret as the fact that the network is robust to noise, obviously, right? So this is stuff that I already said. We also tested this in natural space, so without pre-processing. Take the images, throw them into the architecture right from the scanner, and we get the same performance. So that's also of practical relevance. This is the publication in Neuroimage that if you, in case you were interested in that. And so another example of stuff that we're working on is what I was talking about before, right? A big problem is to separate and stratify syndromes into data-driven categories. And again, this problem only becomes tractable when you talk about neuroimaging if you devise specialized architectures which actually handle, at least in a typical research setting, if you're not Google or Meta, the amount of data that you have to crunch, right? So for this, we used data from the so-called Human Connectome project, and devised an architecture which is able to merge an arbitrary number of types of brain images. We call them modalities. For example, fractional anisotropy, which tells you how much the directionality there is in the cytoarchitecture of the brain, the Jacobi determinant, which I was talking about before, a particular index which tells you how much the neurons are dispersed or aligned within a voxels, and so on and so forth, right? And this architecture is a modification of the conventional unit framework. And here I only have two, in this example, I only have two modalities, but the code is written to accept any number of modalities. And towards through separable convolutions and I guess an architecture which, as you know, has been tuned like we commonly do to solve the specific problems. These modalities get merged and compressed and compressed and compressed until we have a latent space which is only about 1500, which is a vector of length about 1500, right? So we go from 6 or 7 million of numbers to a vector which is about 1500, and we trade, and there's some skip connections here, which have been tuned to perform this task optimally. We're happy also to report that when we actually decode, so when we reconstruct these images, these 3D images, it appears that so these are original images and these are reconstructed images, and of course, one would need a radiologist to appraise that, which were part of the study. But my report that I can give you here is that this is the best ever reconstruction that has been afforded, and it's pretty nice to me to see that obviously these embeddings are mixed, right? So every single modality is being reconstructed with a compression which goes from several millions to 1,000, with a very, very high fidelity, without using adversarial architectures or contrastive learning or anything like that. and yeah, this is just a picture of how the skip connections regularize the loss function landscape in this particular case. So what do we do with this? The point is that now we have these embeddings, and each embedding is an individual and represents in a very compressed manner the brain data from this individual. And so you can go and sort of cluster with various techniques. You can go in and cluster with various techniques these embeddings and see if they actually have any meaning. So you can contrast the clusters that you find. This is a t-SNE visualization of the clusters that we found in this population and their sizes with what will be called phenotypical variables. So other attributes that are available for these patients or for these subjects, which are not part of the training. So are they, what's their intelligence, what's their personality, what's their neuropsychological profile, what's their blood pressure, and so on and so forth. There are about 1,200 phenotypical behavioral variables. And interestingly, we did some data reduction, obviously, on these 1,200 behavioral variables, and we selected some sort of PCA. We used PCA similar to PCA strategies to select components which were related to intelligence, memory, fluid intelligence, which is a different type of intelligence, emotional traits, and so on and so forth, and map them onto these clusters. And I was saying, interestingly, you get some curious results, but they make sense. For example, remember, these are clusters that only come from brain data, and we find, for example, a very, very distinct cluster, which is composed of people which have very high blood pressure. You can see this from the brain, are very high in aggressive traits, are mostly male, are mostly young, are not very smart, intelligence is low, and mostly right-handed, right? So this obviously is a proof of concept, but it makes sense to neurologists and neuroscientists that these traits can be actually discriminated in an unsupervised manner in a cluster that forms from brains that actually belong to a completely healthy population. So the variability is very, very narrow. And so we're able to extract all these clusters. And all these clusters have different profiles that make sense in a neuroscience way. And so obviously, this is the publication. The next step that we're working on is to deploy this and improve this. And I mentioned this, we're improving this with contrastive learning architectures to separate subtypes of disease subtypes, like subtypes of Parkinson's disease, subtypes of Alzheimer's, and any neurological syndrome, really, to figure out whether this baseline prediction and this baseline clusterization will allow us to better target therapeutic strategies according to the neurobiological data that has driven this separation. I talked to you about the graph structure of brain data. So this is another piece of work that I want to mention. Obviously, you're all aware of the fact that there are graph learning operators that can be substituted or in place of traditional convolution operators as feature extractors in whatever kind of architecture you want to use. In this particular work, so this is what I just said. Sorry. So meaning you instead of a regular grid, you have a graph. This graph comes from the brain, and so you will want some type of layer that will extract information about from an irregular topology, and the information is both in the edges and in the nodes of the graph, right? So what we did in this work was, which was done in conjunction with Peter Veljković, who I believe was also a speaker in the seminar series at one point, is to use graph attention networks, which Peter, I'm sure, must have talked about because he was the one who actually designed them, which are very powerful feature extractors in irregular topologies, but we're still not yet completely suited to the problem that we have, because in our neuroimaging data, we don't have one single graph where we want to attend to particular nodes or particular subgraph, but we have one graph per subjects, right? So we have thousands and thousands of brains represented as thousands and thousands of graph, and we want to use them to possibly classify, possibly cluster, possibly, regress against clinically relevant variable that come from the outside of the data domain, right? And so these are the details of the attention networks that were used. I don't know if you're familiar with GATs or graph attention networks. They have a self-attention mechanisms where the weights are defined implicitly, which is able to incorporate both node features and later on also edges, which is important in brain connectivity. And we also used a multi-head attention mechanism in this particular paper. So the part that we had to adapt was the fact that, like I was saying, that we have multiple graphs, right? So the way we did this was to define a master node in each graph. So you have a brain, you have a GAT layer, which extracts features, and this is a master node in each graph, which itself forms a graph, which itself is passed through a graph, through a GAT layer for prediction. So this might seem a little bit cumbersome, but has proved extremely useful, actually, or efficient at least, in solving the problems that we wanted to solve. It's kind of like a graph of graphs, right? To say it in a layman's way. And what did we do with this? We're basically aimed to understand the neural underpinnings of intelligence. So intelligence and predicting intelligence from brain data is an extremely hard problem. There's a lot of smart, no pun intended, smart ways to quantify or to measure intelligence. You can have crystal, fluid, you have composite scores, and so on and so forth. But current attempts to actually quantify it as a function of brain data and predict it as a function of brain data have been basically have sort of failed, right? So what we did here was to use a composite intelligence score, and I have bibliographic references for it here, which was called the G factor, which has been shown to be a better representation of several cognitive abilities, and check whether our multi-graph GAT architecture was able to actually predict the score. And this super simple regression here is basically, it sounds a bit minimalist or maybe like an anticlimax, but actually it's not, because this is basically the whole result of this work, where we are able to predict a subject's intelligence starting only from brain connectivity data, with a correlation between predicted and observed in a test set of about 0.5. And this and an R2 of about 3, 4. Now, this may not seem like much, but like I was saying, if you look in the literature, the maximum that anybody else has ever been able to achieve is a correlation of 0.08, I believe, with an R2 of 0, or similar to 0, mostly negative, right? So this is a very hard problem, but it shows that the information is there, and it shows that by specializing graph layers to using connectivity rather than raw data, you can actually access more higher level information about the brain. Most deep learning brain architectures that you will see in literature actually engage in tasks like sex prediction, right, as a proof of concept. So just to make you aware of the fact that here we are, from neuroscience perspective, on a complexity level which is a little higher than that. Just carrying on along this line of research, what is often done in neuroscience is to use what's called causal connectivity. So, the arrows between the brain regions would have a direction. Which way is the information flowing, from here to here, or vice versa? And the typical paradigm is causality in the Granger sense, which I'm sure you're familiar with, right? A time series or a signal or an entity is said to cause another entity in the Granger sense if the prediction of one entity of the future of one entity can be significantly improved by incorporating the other entity in a prediction model, as opposed to predicting from the past of the first entity exclusively. I hope that was clear enough. So you want to predict X either from the past of X alone or from X and also Y. If this prediction improves significantly, then Y is said to cause X in the Granger sense. Now, how do you actually do this in reality? The vast majority of papers that do this in brain literature are basically just using autoregressive models. So what our contribution here was to substitute these models with specialized recursive neural networks, and in particular echo state networks, which require only random initialization, but very little training because the trainable parameters are not that many. And so both the restricted model, which is this one up here, the one on top, and the unrestricted model, which is the one at the bottom, are formulated in terms of recurrent neural networks. And actually, in the larger model, you see more than one recurrent neural networks because there are some specialization, there's some special heuristics that we employed in making the matrix of the weights block diagonal, and each sub block is an orthonormal matrix, and we've heuristically found that this has increases the detection performance a lot. And in fact, we validated this by using ground truth complex networks, where each node is a chaotic oscillator, and each node is coupled to another node according to directed networks which we generate. This, in particular, this work was done with Duffing oscillators. And here's a simple overall quantification where you can see that this method outperforms, which is the blue one, outperforms all the other available methods in the literature for detecting causal flow between, well, in this case, it would be time series, right? And we call this echo state Granger causality, which is highlighted in red. Incidentally, using this method on real brain data from about 1,000 subjects has allowed us to discover new connections, new directional interactions, which were quite strong and were not necessarily known to the scientific community. So that was a good result for us. Let me carry on. So this is actually a report of work that's ongoing. I thought it might be of interest for this community. We're part of several European consortia, and in one of them, we have been tasked with the slightly science fiction-y idea of generating virtual reality environments from brain activation data. And these virtual reality environments are supposed to, according to the European Commission, reproduce what that object is experiencing and what that object is feeling right in that moment. Because technically, in theory, if we have the technology, this information is encoded in the brain data, right? And so if you have the right translator and the right compression and decompression architecture, you should be able to actually visualize it. And in fact, one of the deliverables of this project is actually re-administering these experiences to the subjects whilst manipulating some of the experiences in a VR environment for a therapeutic usage in major depression. So there's a lot of challenges, as you can imagine, in devising architectures which can do something like this. The assumption here is that the manifold on which the information is represented in the brain can somehow be reproduced in a manifold in our model. At least this is the assumption that we're working with at the moment, so that we can then find a simpler mapping between the two manifolds, right? And then sort of concatenate the transformations. And this is quite challenging. It's quite challenging also because, just to give you an example, the brain has a retinotopic organization, meaning if you show me this object, if you put it here or if you put it here, this is the same object, right? So it should appear in the VR as the same object, but the representation within the brain will actually move, okay? So we already have a spatial invariance problem that we need to build into our architectures, and that's actually what we are working on right now, trying to define kernels that are spatially invariant in this encoder-decoder architecture. Actually, I'm almost, well, I have another 5 or 10 minutes. Other things that we're working on is physically inspired neural networks for physiological systems models. This is, attractive to us as we are a physicist group of physicists working in the Faculty of Medicine, and we do a lot of physiological system models. So physically inspired, or by now they're actually called analytically constrained neural networks, which I find particularly interesting because obviously, as you inject prior knowledge, you will be able to reduce the amount of data that you need to train the network, and that is always an issue in biomedical data. So the loss function in these networks is composed of obviously some sort of a distance function to the data in the training set, but also um, a differential equation normally, or a set of differential equations, which can describe the phenomenon that you are observing. So you need to have an hypothesis about a mechanistic hypothesis about the inner workings of the phenomenon that you're kind of trying to learn. And another part of the loss actually incorporates the boundary conditions, obviously, to these differential equations. So if you manage to do that, you get an architecture which is informed about the physics or the biology or the analytical constraints that you want to build into this model, uses less data and is able to also solve the inverse problem, is also able to actually parameterize the differential equation that describes, the phenomenon that you're studying. And just to show you a quick example of what I actually mean, this here is a set of four ordinary differential equations, which is the Hodgkin and Huxley model for spiking neurons, right? It's very, very typical model and quite complete. It's the starting point for any specialized modeling of neuronal dynamics. And it has a lot of parameters, which are the conductances for every specific ion species that goes through the neuronal membrane and causes neuronal spiking, right? And so what we, as a proof of concept, what I did, this is quite new. I'm starting to work this with one of my students. And so we only have a conference publication on this. But it turned out quite nice because up in graph A, you can see in blue, in red, dashed red, the real spiking data, which comes from a giant squid axon, which is the workbench and test bench for every neurophysiological experiment. And in blue, the predicted spikes by the model. And as you can see, the agreement is excellent for an arbitrary current that's injected into this model. And also the interesting thing is in the table down below that this network is actually able to infer the conductances for every ionic species that's involved in the model with very, very nice accuracy. And since these are dimensional quantities, you can compare them to the literature. And what's most, in a way, surprising, but it's a good surprise, is that these values are in very good agreement with the cumbersome and complicated measurements that have been actually done experimentally in the literature. On top of the fact that this model contains 3 hidden variables that you will never be able to measure, and obviously you can infer these two and therefore understand neuronal dynamics better. And just as a last example of the work that we're doing, very recently, we are kind of thinking about the idea of evolution and how evolution has shaped neuronal networks, so networks of real neurons that you could find in the brains of organisms that live in nature, to perform specific tasks. In other words, if we have a certain neuronal cytoarchitecture, connections, not connections, obviously no brain is a fully connected layer, right? These connections are pruned by evolution and also by growth as an individual becomes an adult. Well, if 300 millions of years of evolutions have done something, that they must have had a point in a way, right? So we are trying to investigate the properties of biological networks, and what I'm showing you here is C. elegans, which is the only organism in nature where a complete neuron by neuron, synapse by synapse mapping has been performed by neuroscientists. So we know this is a little warm, right? It's not a fruit fly this time; it's a worm. And we know that every worm that is born with very, very little inter-individual variability will have 302 neurons with these specific connections. And these connections are, again, very specific. The layout of the neurons is specific. So we're thinking about considerations about energy expenditure and how evolution may have maximized that. This network is a scale-free network, like many networks in nature. So the first step to very simple thing we did was, okay, let's actually try to train this biological network. Apart from on the algorithmic implementation of how to train a network like this, what I can report so far is that it actually, if you compare it to, if you try to do a fair comparison to a simple multi-layer perceptron with the same number of connections and the same number of neurons, it actually performs better in very simple, for now, discrimination tasks like an XOR or discriminating points in space in a two-dimensional spiral or things like that. And it's actually also more robust to dropout, right? And so this is a fascinating, in my opinion, line of research that we'd like to continue on. And the idea is to hopefully one day arrive at a maybe even analytical mapping between the topological properties of these networks, the scale freeness, the exponents, and so on and so forth, and things like their training efficiency or robustness or anything like that. And I think this is a very, very quick overview of what I wanted to speak to you today. Thank you very much for your attention. I'll be happy to answer any questions you have.
Okay, thanks, Nicola, for this very, very interesting and broad and depth overview of your research results. They're very interesting and have an actual impact, as we mentioned before the seminar went online, on actual real life problems. And with respect to that, I am having a look to see if there's any question coming from the YouTube community. You can also ask questions, and I have questions on my personal question on the representation of the graph structure of brain data. I mean, what is the impact of the evolution of technology on better understanding and reconstructing and analyzing actual complex brain.
So you mean technology for retrieving brain data, or technology in the sense of computational architectures? Sorry for the counter.
Both.
Okay.
Both.
Okay. Yeah. So that's a good question, because deriving an adjacency matrix from a connectivity matrix from a brain is, and how to do that best is an active area of research in the first place. 10 years ago, people started by doing a functional MRI, so brain activity in time, parcellate, time series, and just do pairwise Pearson's correlation. This is how it all started, right? And then obviously that introduces redundancy, and then people started using multivariate models or autoregressive models, which at least eliminate redundancy, partial correlations, and so on and so forth. There's two things. The imaging and the data extraction technology is extremely important, especially because of signal-to-noise ratio, because the scale at which you extract the information is crucial, and we can, at the moment, we cannot really go below sort of a 2mm cubed spatial resolution, whereas ideally you would like to do something like calcium imaging or optogenetics, which is something you can only do in animals, and go at the neuron resolution. So as fields become higher and higher and new technologies come, that will increase the SNR. But also I have to say, I didn't mention this in the seminar, but in my opinion, the way that these architectures, talking on the computational and the algorithmic side, should actually progress is to actually learn the adjacency matrix as part of the classification process rather than pre-computing it. And this is something that we're working on also in terms of dynamic brain community detection, because obviously otherwise you're going to introduce and we have, anybody who does connectivity actually knows this, although they never put it in the publication. You're going to introduce a huge bias in your results. You can, if you change the method in which with which you design your adjacency matrix, you basically can tune the classification results as much as you want. So this is actually a real issue in the neuroscience literature right now. I hope I answered your question.
Perfect. I think there's Massimo waiting to be.
Hello.
Okay. Thanks,
Hello, Massimo.
Nicola.
Please go on.
Okay, thanks, Nicola, for very
Don't
nice.
stop.
I can hear him. Massimo is here.
Okay, can you hear me? Nicola, can you
yes.
hear? Okay.
Yes.
Thank you very much. A very nice talk. In some sense, you already answered the question that I'm going to ask, but again, I would like to ask this somehow provocative question. You present a very interesting number of applications that somehow rely on the, say, hidden assumption that you have, say, reliable data, that your input data are reliable. I'm not an expert, but I've been working with, say, MRI management or images in the last 2 years. And I am under the impression that most of the existing software was developed, say, a couple of decades ago with different purposes, with different tools. And now many people want to use the same software for completely different purposes, like personalized medicine. And I see a huge problem there in image quality, in the preprocessing, what you call preprocessing. Do you think that some specific work based on neural network, based on modern method, could improve the quality of this preprocessing? And do you see this as an interesting research direction, or it's too boring, it's the back office?
No.
So
No,
I understand that it's somehow provocative, but I would really get your opinion.
Thank you for the question. It's not. I mean, it's very relevant and it's not provocative, and it's an issue. So let me tell you let me start quickly by telling you you're perfectly correct. In fact, there are people in the community, in the sort of deep learning community for imaging, that have basically concluded after so many years that fMRI data is 80% noise. Right, and so a linear ridge regression will always perform better than a deep graph neural architecture. Right. So, and I don't personally agree, but I can see where they're coming from, and where they're coming from is the challenges that you are highlighting here. We have low signal to noise ratio. We have a pre-processing which is definitely doesn't exploit all the features of the data. So SNR is something that needs to come from technology, right? And not necessarily from, although, and this is where I wanted to answer your last part of the question, there is a lot of development in neural network architectures, not only to improve image quality, but even at the stage before, at the acquisition step, so to design and to guide as you know, MRI images are acquired in Fourier space, right? So which is called K-space. So to guide the trajectory that you take within the K-space, and also to reconstruct images. So there are today only research, unfortunately, applications yet that actually take the K-space and generate sort of physical space images that are much better quality than the actual reconstruction software that is given by the vendors. And this is even before the preprocessing. There are neural architectures that have been able to solve ill-posed problems, so that you can severely undersample the K-space, which is a huge benefit because the patient will stay there for 7 minutes instead of 70 minutes. and still give you the same quality image. There are neural architectures which are able to reconstruct images which you will only get by injecting contrast media. So with all the problems that
side effect, yeah,
that even radioactive contrast media from images that have been acquired in the same patients without contrast media. So it's not boring at all. In fact, it's a huge area of research, especially in the field of again, I guess, generative model, but also contrastive imaging. And I'll say this very last thing. There's the first company that actually is offering the service has just gotten FDA approval in the US. They are generating a nuclear imaging just from MRI. So they take non-ionizing radiation and they in the raw case space and they give you the equivalent of a PET scan, right? So it's quite exciting development. Actually. Again, I tend to talk a lot. I don't know if I answered your question.
I will contact you offline. Very interesting.
Okay.
I will contact David about that. Thank you very much, Nicola. Once again, thank you very much.
Thank you for
very nice talk.
Okay, let me have a look if there's some other question from the YouTube audience. No, I mean, I believe the Massimo hit the right spot in the. I mean, one of the main issues behind analyzing and studying and modeling the brain and in general living body data. So that this is also the signal to noise ratio. I mean, the problem of having reliable data is vital to any kind of.
Yes, and I will say this. I will say that the sort of only applied community is, in my opinion, although this is broadcast on YouTube, but okay, it's maybe not completely aware of how important this is, even with conventional and not neural network based analysis. So only now papers are starting to appear in Nature Neuroscience, and so on and so forth, which say, hold on, guys, if you use this model, you're going to have 70% false positives. If you use this number of subjects, you're basically producing results that are not reproducible or have even a high intra-individual variability, which is, so if you scan and rescan the same subjects, which is the same as the variability between two other subjects, and so on and so forth. So that's actually a very, very hot topic right now. But again, this is the real brain data. So it is at the interface between what a computer scientist would ideally like to have, or a mathematician, and what a medical doctor is able to provide. So it's exciting to work at this interface. Definitely,
it's exciting because in the end, I mean, the outcome is it saves lives, or it can help save lives, or may improve the quality.
We hope.
It has really,
we hope you hope to give some contribution. Yes.
Yeah. Okay, so let's thank again this very, very interesting speaker.
Thank you for having
seminar, Nicola. It has been a pleasure having you as a guest for this seminar. I will I give some pointers. Next week there will be the Cnr-Iac seminars on Wednesday, and we will continue the artificial intelligence and mathematics seminar series in the weeks to come. Have a look at the web for news on the following seminars. Thank you, everybody, and see you soon online.